Good morning. Welcome to the class, students. In the previous class, we finished theory of equations. The remaining part of theory of equations is deleted by the Board of Intermediate. So now I come to the chapter permutations and combinations. So this is a permutations and combinations. Okay, this is this chapter is of a different kind. So when we compare with the other chapters, it is of a different. So in general, one can think of that mathematics is just a step-by-step procedure, whereas this permutations and combinations is very interesting, but it is typical. We have to understand the things clearly. That is the major applications of permutations and combinations are in day-to-day life applications and in theory of probability. So we have many applications in these fields. Okay. In day-to-day life, suppose, for example, if we go to the hotel, so we have to select some items from the menu card. Okay, so how many ways can we select the menu from the menu card? Okay, that is we count the number of ways of performing a work without explicitly doing that work. That is the major technique here. So ఒక పని చేయకుండానే ఆ పనిని ఎన్ని విధాలుగా మనం చేయవచ్చు అనేటువంటి అంశాన్ని మనం లెక్కిస్తామన్నమాట. సో దట్ ఈస్ ద బ్యూటీనెస్ ఆఫ్ దిస్ చాప్టర్. ట్రై టు అండర్స్టాండ్ ఎవ్రీథింగ్. ఓకే, సో ఇక్కడ ఏంటంటే అండ్ మోర్ ఓవర్ ఫస్ట్ ఆఫ్ ఆల్ ఐ డిఫరెన్షియేట్ బోత్ ది థింగ్స్. That is permutations and combinations. Permutation is nothing but it is an arrangement. Okay. Combination means it is a selection. Okay. So మన దగ్గర 10 వస్తువులు ఉన్నాయి. 10లో నుంచి ఒక 6 వస్తువులు సెలెక్ట్ చేసుకోవాలి, ఎన్నుకోవాలి. సో ఎన్ని విధాలుగా 10 వస్తువుల నుంచి 6 వస్తువులను ఎన్నుకోగలరు, సెలెక్ట్ చేసుకోగలరు. అంటే సెలెక్ట్ చేసుకొని 6 వస్తువులను తీసుకొని ఒక చోట మనం పెట్టేసుకుంటాం అన్నమాట. దట్ ఈస్ కాల్డ్ కాంబినేషన్. సో వేరాజ్ ఇన్ పర్ముటేషన్స్ వీ హ్యావ్ 2 వర్క్స్. సో ఫర్ ఎగ్జాంపుల్ మీకు ఇచ్చిన 10 వస్తువుల నుంచి 6 వస్తువుల్ని మీరు సెలెక్ట్ చేసుకొని వాటిని ఒక రోలో అరేంజ్ చేయాలి. అంటే ఒక 6 బ్లాంక్ ప్లేసెస్ ఉన్నాయి. వాటిలో మీరు సెలెక్ట్ చేసుకున్నటువంటి 6 వస్తువులను అరేంజ్ చేయాలి. అంటే ఇక్కడ 2 పనులు ఉన్నాయి. ఒకటి 10 వస్తువుల నుంచి 6 వస్తువులు మీరు సెలెక్ట్ చేసుకోవాలి, అది ఫస్ట్ చేయాలి, ఫస్ట్ వర్క్. అండ్ దెన్ ఈ మీరు సెలెక్ట్ చేసిన 6 వస్తువుల్ని ఇక్కడ 6 ప్లేసెస్లో మీరు అరేంజ్ చేయాలి. ఓకే, ఇక్కడ ఏమవుతుందంటే సెలెక్షన్ అండ్ arrangement. So that is in the case of permutations. So that should be very clear. Whenever a problem is given to you, then you have to understand the things which is given in the problem, and what we have to do is that you have to explicitly understand clearly, and then you have to come to conclusion whether permutations are to be taken or combinations are taken. That you have to understand clearly. So okay, it is very easy when you understand. Try to follow everything whatever I am explaining here. Okay, so first of all I begin with a certain denotation. Okay, na? So this is a small introduction about permutations. The permutations and combinations are I have several applications in statistics, in probability, everything, somewhere in some engineering branches, etc. we use. So see here, if n is a positive integer, see here a small denotation is given, positive integer. The then n factorial already మీకు తెలుసు ఈ చిన్న డివోషన్ again I am explaining. n factorial is n into n minus 1 n minus 2 into 3 to 1 1 దాకా n పాజిటివ్ ఇంటీజర్ కాబట్టి n లో నుంచి ఒక్కొక్కటి మనం సబ్ట్రాక్ట్ చేస్తూ కాన్సిక్యూటివ్ ఇంటీజర్స్ ప్రోడక్ట్ ఫ్రమ్ 1 to n, that is n factorial. సో బై కన్వెన్షన్ ఇక్కడ 0 factorial అంటే మనం 1 అని తీసుకుంటాం. ఓకే, ఇట్ ఈస్ జస్ట్ కన్వెన్షన్. 0 factorial is 1, 1 factorial 1, 2 factorial 2 into 1, 3 factorial 3 into 2 into 1, వెరీ సింపుల్. 3 factorial 3 నుంచి 1 దాకా వచ్చేవన్నీ మల్టిప్లై చేయాలి. అలాగే 4 నుంచి 1 దాకా. ఇక్కడ ఏం చేశాను, 4 factorial ఉన్నప్పుడు 4 into ఇదేమవుతుంది 3 factorial, సో 24. 5 factorial ఉన్నప్పుడు 5 into 4 into 3 into 2 into 1, that is 5 into 4 factorial. ఓకే, ఇలా 6 factorial, 6 into 5 factorial. ఇన్ జనరల్ n factorial అంటే ఎలా రాయొచ్చు, n into n minus 1 factorial, n into n minus 1 factorial. ఆర్ మనం ఎన్ని నెంబర్స్ కావాల్సింటే అన్ని నెంబర్స్ రాసి ఆ తర్వాత n minus 2 factorial రాస్తే సరిపోతుంది. ఓకే, సో దిస్ ఇస్ about factorial of n. So okay, now I come to the concept. So before going to enter into the detailed discussion, we have a small concept, that is fundamental principle of counting. So this chapter permutations and combination is all about counting. See, it is nothing but all about counting. So we count how many ways this work can be done, how many ways we can select, how many ways we can arrange. These are the major work what we are going to do. Okay, try to understand. And here I see fundamental principle of counting. Fundamental principle of counting. So okay, count చేయడానికి ఒక ఫండమెంటల్ ప్రిన్సిపల్ చెప్తుంది. ఏంటి అది అంటే ప్రాథమిక సూత్రము. ఓకే, తెలుగులో చెప్పాలంటే see here, if your work W1 can be performed in m different ways and another work W2 can be performed in n different ways, then the two works one after the other can be performed in mn ways. This is given by our fundamental principle of counting. So what is happening is ఒక వర్క్ అంటే ఏ వర్క్ అని మీరు డౌట్ పడాల్సిన అవసరం లేదండి. వర్క్ అంటే అది ఏదైనా కావచ్చు, అంటే సెలెక్టింగ్ ద థింగ్స్, అరేంజింగ్ ద థింగ్స్ అండ్ ఓకే గోయింగ్ ఫ్రమ్ హియర్ టు దేర్. సో ఏ వర్క్ అయినా కావచ్చు. ఇఫ్ యువర్ వర్క్ W1 కెన్ బి పర్ఫార్మ్డ్ ఇన్ m డిఫరెంట్ వేస్, దాన్ని m విభిన్న విధాలుగా చేయవచ్చు. అండ్ అనదర్ వర్క్, ఇంకొక వర్క్ W2 కెన్ బి పర్ఫార్మ్డ్ ఇన్ n డిఫరెంట్ వేస్. సో ఈ W2 సెకండ్ వర్క్ W2 కెన్ బి పర్ఫార్మ్డ్ ఇన్ n డిఫరెంట్ వేస్. ఓకే, సో దెన్ ద టూ వర్క్స్ ఎలా చేయాలి, సైమల్టేనియస్గా, వాళ్ళు అందుకే రాసచ్చు వన్ ఆఫ్టర్ ద అదర్ అంటే ఒకదాని తర్వాత ఒకటి చేసిన తర్వాత వెంటనే ఇంకొకటి, నాట్ సైమల్టేనియస్. ఓకే, సక్సెసివ్లీ వన్ ఆఫ్టర్ ద అదర్. సో కెన్ బి పర్ఫార్మ్డ్ ఇన్ mn వేస్. mn అంటే ఇక్కడ m into n వేస్, అంటే ఒక పనిని m విభిన్న విధాలుగా చేయొచ్చు. ఇంకొక పనిని W2 అని ఇంకొక పనిని n విభిన్న విధాలుగా చేయొచ్చు. అలాంటప్పుడు మొత్తం రెండు పనుల్ని ఒకదాని తర్వాత ఒకటి mn విభిన్న విధాలుగా చేయొచ్చు అని చెప్పి ఈ ఫండమెంటల్ ప్రిన్సిపల్ ఆఫ్ కౌంటింగ్ చెప్తుందండి. సో దీన్ని మేజర్గా మనం ప్రతి ప్రాబ్లంలోనూ సో ఈ ప్రిన్సిపల్ ఏదైతే చెప్తుందంటే 2 మల్టిప్లై చేయమని చెప్తామండి. సో దీన్ని బేస్ చేసుకుని దీన్ని వాడుకొని మనం చాలా చోట్ల ప్రాబ్లమ్స్ చేస్తాము, అది మీరు గుర్తుంచుకోవాలి. ఓకే, సో దీని ప్రూఫ్ ఎవ్రీథింగ్ మనం లేదు. ఐ అవాయిడెడ్ ఆల్ దోస్ థింగ్స్. ట్రై టు అండర్స్టాండ్ జస్ట్ వాట్ ఐఎమ్ ఎక్స్ప్లెయినింగ్ హియర్. సో ఓకే, నెక్స్ట్ ఫర్ దిస్ I have given a small example here. So if a girl has 4 skirts, ఒక గర్ల్కి then ఉన్నాయి 4 skirts ఉన్నాయి, S1, S2, S3, S4 and 5 tops, T1, T2, T3, T4, T5. Then she can select a pair to wear in హౌ మెనీ వేస్ 4 into 5 దట్ ఈస్ ఈక్వల్ టు 20 వేస్ అని చెప్పి చెప్తున్నాను. అంటే ఒక బాలికకి ఎన్ని ఉన్నాయి, 4 స్కర్ట్స్ ఉన్నాయి. ఎన్ని, S1 S2 S3 S4 అండ్ 5 టాప్స్ T1 T2 T3 T4 T5 మామూలు స్కర్ట్స్ మనకు ఉంటాయి కదా, బ్లూ గ్రీన్ రెడ్ ఏవో కలర్స్ డిఫరెంట్ టైప్స్ డిఫరెంట్ స్కర్ట్స్ ఈ విధంగా 4 స్కర్ట్స్ 5 టాప్స్ ఉన్నాయి. సో మనము వేసుకోవడానికి అమ్మాయికి ఎన్ని కావాలి, ఒక స్కర్ట్ ఒక టాప్ కావాలి. కాబట్టి షీ కెన్ సెలెక్ట్ హౌ మెనీ వేస్. ఇప్పుడు ఫర్ ఎగ్జాంపుల్ ఇక్కడ మీకు చూపించాలంటే S1 స్కర్ట్ S1 తీసుకున్నప్పుడు టాప్ ఏదైనా వేసుకోవచ్చు. S1 T1 S1 T2 S1 T3 S1 T4 ఇలా. సో ఎన్ని వచ్చాయి, S1కి మొత్తం 5 వచ్చాయి. S1 అంటే S1 ఒకటి తీసుకున్నప్పుడు 5 వచ్చాయి. మళ్ళీ S2 తీసుకున్నప్పుడు మళ్ళీ అగైన్ S2 T1 S2 T2 S2 3T మళ్ళీ 5 వస్తాయి. మళ్ళీ S3కి ఇలాగే 5 వస్తాయి. మళ్ళీ S4కి 5 వస్తాయి. సో 5 4 20 ఈ విధంగా మనం కౌంట్ చేయొచ్చు. వితౌట్ ఎక్స్ప్లిసిట్లీ రైటింగ్ ఆల్ దీస్ థింగ్స్, okay, we can count how many ways she can select a pair to wear. Okay, that is the beautiness of these permutations. Okay, permutations and combinations. What I am telling, please observe. Without writing all the arrangements, all the possibilities ఈ విధంగా మనం లిస్ట్ అవుట్ చేయాలి. సపోజ్ ఫర్ ఎగ్జాంపుల్ ఈ స్కర్ట్స్ కానీ టాప్స్ కానీ సో మెనీ నెంబర్ లార్జర్ నెంబర్ ఉన్నాయనుకోండి, అప్పుడు రైటింగ్ ఆల్ దీస్ ఎక్స్ప్లిసిట్ ఆల్ దీస్ అరేంజ్మెంట్స్ పేర్స్ అన్నీ రాయాలి, చూపించాలి అని అంటే సో ఇట్ ఈస్ ఏ టీడీఎస్ వర్క్, ఇట్ ఈస్ ఎ డిఫికల్ట్ క్లాస్ వర్క్స్ వర్క్. సో అవన్నీ ఎక్స్పెక్ట్ చేయం మనం. సో జస్ట్ we say how many ways she can select a pair, so that all possibilities we can find the number, we can find. Okay, 4 into 5 20 ways. Okay, this is the magic of permutations. How many ways can that work can be performed. Okay, this is okay. A work W1 can be performed. W1 and ఇక్కడ selecting a skirt, that can be performed in 4 ways. Selecting a top, that can be performed in 5 ways. అంటే టాప్ తీసుకునేటప్పుడు T1 తీసుకోవచ్చు, T2 T3 T4 T5. ఓకే, ఇలా 5 ways గా చేయొచ్చు. సో ద టోటల్ వర్క్ కెన్ బి డన్ ఇన్ mn ways, m into n ways, 4 into 5 ways. Okay, that is the example what I have given to you. Next, after this we have a permutation. I am going to define permutation. Linear, permutations lo rendu rakalu, 2 types of permutations. One is linear permutation and another is circular permutation. Okay, linear and circular. ఈ లీనియర్ సర్క్యులర్ అంటే ఫస్ట్ నేను లీనియర్ గురించి చెప్తాను, తర్వాత సర్క్యులర్ మాట్లాడతాను. సో ఫ్రమ్ ఏ గివెన్ ఫైనైట్ సెట్ ఆఫ్ ఎలిమెంట్స్ సిమిలర్ ఆర్ నాట్, అంటే మీకు ఒక ఫైనైట్ సెట్ ఆఫ్ ఎలిమెంట్స్ ఇచ్చాడు. దే మే బి సమ్ ఆర్ సిమిలర్ ఆర్ సమ్ ఆర్ డిఫరెంట్ ఆర్ ఆల్ ఆర్ డిఫరెంట్. సో హౌఎవర్ దే మే బి. సో నీకు ఒక ఫైనైట్ సెట్ ఆఫ్ ఎలిమెంట్స్ ఇచ్చాడు. ఇచ్చిన తర్వాత సెలెక్ట్ సమ్ ఆర్ ఆల్ ఆఫ్ దెమ్. వాటి నుంచి ఏం చేయాలి, సెలెక్ట్ సమ్ ఆర్ ఆల్ ఆఫ్ దెమ్. అందులో కొన్నైనా తీసుకోవచ్చు లేదా అన్నైనా తీసుకోవచ్చు. అంటే అరేంజ్ దెమ్ ఇన్ ఏ రో లైన్. అంటే మనం సెలెక్ట్ చేసుకున్నటువంటి వస్తువులు ఏవైతే ఉన్నాయో వాటిని వీ హ్యావ్ టు అరేంజ్ దెమ్ ఇన్ ఏ రో వరుసలో లైన్లో అరేంజ్ చేయాలి. సో ఇది మనం చేయాలి. సో చేసినప్పుడు సచ్ అరేంజ్మెంట్, ఆ విధంగా చేయగా వచ్చినటువంటి వన్ అరేంజ్మెంట్, సచ్ అరేంజ్మెంట్ ఈస్ కాల్డ్ ఏ లీనియర్ పర్ముటేషన్, ఈస్ కాల్డ్ ఏ లీనియర్ పర్ముటేషన్ or simply we call it as permutation. Linear or generally we call it as permutation. Permutation means such arrangement. అంటే కొన్ని వస్తువులు ఇచ్చారు, వాటిలో అన్నింటిని కానీ లేదా కొన్నింటిని కానీ తీసుకొని ఆ తీసుకున్నటువంటి వస్తువులు ఒక లైన్లో ఒక వరుసలో అరేంజ్ చేసినట్లయితే ఈ వచ్చిన ఈ అరేంజ్మెంట్ని పర్ముటేషన్ అంటారు. ఓకే, దట్ ఈస్ కాల్డ్ లీనియర్ పర్ముటేషన్ ఆర్ పర్ముటేషన్. ఓకే, so this is the definition of a linear permutation. So for this I am giving one small example here. See here, from the letters of the word moral. From the letters of the word moral. Moral అనేటువంటి ఒక వర్డ్ ఇచ్చారు మీకు. సో 3 letter permutations are 3 letter permutations అంటే మోరల్ అనేటువంటి ఈ వర్డ్లో ఎన్ని ఉన్నాయి? 5 letters ఉన్నాయి. ఈ 5 letters-లో 3 letter permutations are. 3 తీసుకొని పర్ముటేషన్ అరేంజ్ చేయాలి. సో 3 మన ఇష్టం, ఎలా అయినా తీసుకోవచ్చు. చూడండి, ఎంఆర్, ఎంఆర్, ఐఏఎన్ఆర్, మోల్, ఎంఓఎల్, ఎంఆర్ఈ. ఓకే, ఈ విధంగా ఇలా ఎన్నైనా మనం రాసుకోవచ్చు. ఓకే, సో what we are going to learn in this chapter is how many such arrangements are, such permutations, how many permutations do we get, that we are going to learn now. Okay, so try to understand. Okay na? Here I am giving one more example for permutation, linear permutation. See here in the previous example we will take letters of the alphabet. Here using the digits, we use the digits. Using the digits 1, 2, 3, 4, 5, 2 digit permutations are. So మనకి 5 digits ఇచ్చారు. 1, 2, 3, 4, 5 digits లో మనం 2 digit permutations అంటే 2 తీసుకొని రెండు కావాలంటే రెండింటిని సెలెక్ట్ చేసుకోవాలి. చెప్పండి, ఇప్పుడు చూడండి, ఈ 1, 2 సెలెక్ట్ చేసాం అనుకోండి, 1, 2 ఒక పర్ముటేషన్, 2, 1 ఇంకో పర్ముటేషన్. సో 1, 2, 2, 1 ఇక్కడ మీరు అర్థం చేసుకోవచ్చు, సెలెక్షన్ అండ్ అరేంజ్మెంట్. సో ఓకే, 2 digits నేను ఫస్ట్ సెలెక్ట్ చేశాను అంటే ఇక్కడ 5 digitsలో 2 సెలెక్ట్ చేశాను, 1 and 2. వీటిని అరేంజ్ చేస్తున్నాను రోడ్. సో 1, 2 రెండింటిని ఎలా అరేంజ్ చేయొచ్చు? 1, 2, 2, 1. ఓకే, సో ఈ విధంగా 1, 2, 2, 1, 2, 3, 4, 5, 3, 4, 1, 5. So we can get many 2 digit permutations like this. So we can without explicitly writing all these 2 digit permutations, we can say how many 2 digit permutations can we get. So that is given in this chapter. Okay, so these are the some examples for a permutation. So now I come to the next item, that is permutations of n dissimilar things taken or at a time. Permutations of n dissimilar things taken or at a time. That is, n dissimilar things are given. What n dissimilar things నుంచి r వస్తువులు, r things, r తీసుకొని permutations చేయాలి. Permutations we have to count the number of r permutations, r digit permutations. So see here, let n and r be positive integers and r is less than or equal to n. r is less than or equal to n ఎందుకంటామంటే n వస్తువుల నుంచి r వస్తువులు తీసుకుంటాం. ఓకే, n వస్తువుల నుంచి r వస్తువులు తీసుకుంటామంటే n should be greater than or equal to r, and I mean r should be less than or equal to n. Okay, so that is what it is. So okay, the number of permutations of n dissimilar things taken or at a time is, so n number of permutations of n dissimilar things, n విభిన్న వస్తువులు నుంచి r వస్తువులు తీసుకోగా వచ్చే పర్ముటేషన్స్ ఎన్ని, నెంబర్ ఆఫ్ పర్ముటేషన్స్. అంటే నేను ఇందాక మీకు ఎగ్జాంపుల్ చెప్పాను, 10 విభిన్న వస్తువులు ఇస్తే 10 నుంచి 6 తీసుకొని అరేంజ్ చేయాలి. సో ఆ విధంగా ఎన్ని మొత్తం అరేంజ్మెంట్స్ ఎన్ని వస్తాయి అని చెప్పి సో మనం కౌంట్ చేయడానికి ఈ థీరం మనకి యూజ్ చేస్తుంది. See here once again, so the number of permutations of n dissimilar things taken or at a time. n విభిన్న వస్తువుల నుంచి 6 వస్తువులు తీసుకోగా వచ్చే పర్ముటేషన్స్ ఎన్ని అంటే n into n minus 1, n minus 2 and so on, n minus r minus 1. This can be written as n minus r plus 1 also. So ఇది ఈ థీరం చెప్తుంది, n into n minus 1, n minus 2 and so on, n minus r plus 1. So this is the theorem. But the proof, the theoretical proof of this theorem is deleted from the syllabus. So that's why just I am giving a clear idea about the proof of this theorem. Okay, just follow this what I am explaining here. So చూడండి n విభిన్న వస్తువులు ఇక్కడ ఉన్నాయి మన దగ్గర. So see here, so this is the set of n dissimilar things. n dissimilar things ఇక్కడ ఉన్నాయి. So వీటి నుంచి మన we have to select r things and we have to arrange those r things in r blank or places. That is called one permutation. Okay na? So అయితే ఈ చెప్పినటువంటి ఈ పని చేయడానికి దీన్ని ఎలా చేస్తున్నానంటే see here, మనం r వస్తువులను అరేంజ్ చేయాలి కాబట్టి I have taken r blank places. See here, first place, second, third, fourth and so on r blank places are here. Okay, so ఇప్పుడు ఈ I have to fill this first blank place using this n dissimilar things. Okay, I have to fill this first blank place using these n dissimilar things. Okay, ఎలా చేస్తామంటే ఈ ఫస్ట్ బ్లాంక్ ప్లేస్ లో ఎన్ ఉన్నాయి, ఎన్లో అంటే ఎన్ ఒకటి పెట్టచ్చు, ఒక వస్తువు పెట్టచ్చు, రెండో వస్తువు, మూడో వస్తువు. సో ఏదైనా అంటే హౌ మెనీ వేస్ యూ కెన్ ఫిల్ దిస్ బ్లాంక్ ప్లేస్? n ways, because n are dissimilar things. ఓకే, నీ దగ్గర 4 వస్తువులు ఉన్నాయి, 4 వస్తువులతో ఈ ప్లేస్ని ఫిల్ చేయాలంటే హౌ మెనీ వేస్ యూ కెన్ ఫిల్? 4 ways. మొదటిది పెట్టచ్చు, రెండోది పెట్టచ్చు, మూడోది, ఈ విధంగా. సో మన దగ్గర n ఉన్నాయి కాబట్టి n వస్తువులతో ఈ ఫస్ట్ బ్లాంక్ ప్లేస్ని వీ కెన్ ఫిల్ ఇన్ n different ways. ఓకే, after filling the first blank place, the second blank place. So how many ways? So because of one thing is out of n different things, one thing is used here. So how many things are remaining? You will have n minus 1 different things. So వాటితోటి మీరు సెకండ్ బ్లాంక్ ప్లేస్ మనం ఫిల్ చేయాల్సి ఉంటుంది. హౌ మెనీ వేస్? n minus 1 ways. ఓకే, మళ్ళీ సిమిలర్లీ థర్డ్ బ్లాంక్ ప్లేస్. సో మన దగ్గర రెండు యూజ్ చేసాం, ఫస్ట్ బ్లాంక్ ప్లేస్లో ఒకటి, సెకండ్ బ్లాంక్ ప్లేస్లో ఇంకోటి. సో హౌ మెనీ థింగ్స్ ఆర్ లెఫ్ట్ ఓవర్? n minus 2 things. Using these n minus 2 things, you will have to fill third blank place. How many ways? n minus 2 ways. We can fill the third blank place in n minus 2 different ways. Similarly, fourth blank place. Alaga and so on. This is the fourth blank place. We can guess how many ways? n minus 1, third n minus 2, fourth n minus 3. So fourth blank place can be filled in n minus r minus 1 ways. n minus r minus 1 ways. Okay, so according to the fundamental principle of counting, so the total work, so w1 w2, ఇది fundamental principle of counting అనేది ఓన్లీ టూ వర్క్స్ w1 w2కే కాదండి, అలా 1 by 1 ఎన్ని వర్క్స్ కైనా కూడా మనం అప్లై చేసుకోవచ్చు. సో దట్స్ వై, సో ద టోటల్ వర్క్ కెన్ బి done in n into n minus 1, n minus 2, n minus 3 and so on, n minus r minus 1 ways. So that is the total work. అందుకని ఇక్కడ వచ్చింది చూడండి. సో మొత్తం అన్ని మల్టిప్లై చేయండి, n into n minus 1 into n minus 2 and so on, n minus r minus 1 ways. So this is the number of permutations of n dissimilar things taken or iterated. This is once again, this is the number of ways of filling r blank places using n dissimilar things. Okay, that is same as the number of permutations of n dissimilar things taken or iterated. Once again, you have to understand two things here. So one is the number of permutations of n dissimilar things taken or at a time is equal to the number of ways of filling or blank places using n dissimilar things. That is the first one. Okay, second, later, so in r blank places by using the fundamental principle of counting, how many ways can you fill? So this is n into n minus 1 into n minus 2 into n minus r minus 1. That's all. Okay, So this is this theorem, try to understand. Okay, next we have a small denotation. We denote the number of permutations of n dissimilar things taken or at a time is NPR. సో n విభిన్న వస్తువుల నుంచి r వస్తువులు తీసుకోగా వచ్చేటువంటి పర్ముటేషన్స్ ఏమి నెంబర్ ఆఫ్ పర్ముటేషన్స్ ని NPR అనే డినోషన్ తోటి మనం డినోట్ చేస్తాము. సో నెంబర్ ఆఫ్ పర్ముటేషన్స్ ఆఫ్ n డిసిమిలర్ థింగ్స్ టేకెన్ ఆర్ ఎట్ టైం. సో ఈస్ NPR. ఈ n ఇదేమో మొత్తము ఎన్ని థింగ్స్ డిఫరెంట్ థింగ్స్ ఉన్నాయనేది ఇది చూపిస్తుంది. సో ఇప్పుడు ఫర్ ఎగ్జాంపుల్ 5లో ఉన్నాయి, 5లో నుంచి 3 వస్తువులు తీసుకోవాలి. 5p3 we write. So 5p3 means the number of permutations of three things of 5 dissimilar things taken 3 at a time. 5 వస్తువుల నుంచి 3 వస్తువులు తీసుకోగా వచ్చే పర్ముటేషన్స్ ఎన్ని అని ఇది చెప్తుంది. ఓకే, ఇది డినోషన్ NPR. ఓకే, NPR. సో NPR అంటే ఇక్కడ NPR ఇక్కడ మనం అకార్డింగ్ టు దిస్ థియరం వాట్ ఈస్ NPR? n into n minus 1, n minus 2 and so on, n minus r minus 1. ఇది పైన మనం ఇందాక ప్రూవ్ చేసాం. ఓకే, సో r బ్లాంక్ ప్లేసెస్ ని హౌ మెనీ వేస్ ఫిల్ చేస్తామంటే మనకి ఇది వచ్చింది. దిస్ ఇస్ నథింగ్ బట్ ద నెంబర్ ఆఫ్ పర్ముటేషన్స్ ఆఫ్ n dissimilar things taken or at a time. సో దీన్ని మనకి ఇక్కడ ఈ మొత్తం ఇలాగే ఉంచాను. ఇక్కడ చూడండి n minus 1, n minus 2, n minus 3, n minus r minus 1, నెక్స్ట్ n minus r, నెక్స్ట్ n minus r minus 1. ఇలా మనం చివరి దాకా వెళ్తే 3 into 2 into 1 వస్తుంది. సో ఈ మొత్తం టర్మ్ని ఇక్కడ ఇంటూ మల్టిప్లై చేశాను అండ్ అదే టర్మ్ని బై చేశాను. ఓకే, సో n minus r minus 1 వరకు ఉంది. నెక్స్ట్ వచ్చేదంటే n minus r minus 1. సో ఇంకా డిక్రీజ్ అయిపోతుంది 1 by 1. సో ఇక్కడ మీరు న్యూమరేటర్ గమనించినట్టయితే n into n minus 1, n minus 2 and so on, n minus r minus 1, n minus r, n minus r ఇలా 3 to 1 దాకా ఉంది. కాబట్టి the numerator is nothing but n factorial and the denominator is n minus r, n minus r minus 1, n minus r minus 2 and so on 3 to 1. That is nothing but n minus r factorial. So therefore NPR is equal to n factorial by n minus r factorial. NPR is equal to n factorial by n minus r factorial. This you have to remember as a formula. And also ఇక్కడ ఇది కాకుండా NPR ని ఎలా చెప్పాలంటే NPR ఇక్కడ దిస్ ఇస్ NPR కదా. సో ఈ NPR ని ఎలా చెప్పాలంటే the product of consecutive integers beginning with n. The product of all consecutive integers beginning with n. n తో స్టార్ట్ చేసి r కాన్సిక్యూటివ్ ఇంటీజర్స్ ప్రోడక్ట్ చేసినట్టయితే మీకు NPR వస్తుంది. ఫర్ ఎగ్జాంపుల్ ఇక్కడ మీకు చూపిస్తాను. 5p3 5తో మొదలుపెట్టి 3 r అంటే ఎంత 3 రాయాలి. 3 product of 3 consecutive integers. So this is 5p3. Okay. See students, our NPR is equal to n into n minus 1, n minus 2 and so on, n minus r plus 1. Just now in previous theorem we proved this, that is NPR means the product of r consecutive integers beginning with n into n minus 1. అలాగా r నెంబర్స్ మనం ప్రోడక్ట్గా చేస్తాం. సో దీన్ని ఇలా కూడా రాయొచ్చు. జస్ట్ ఇంప్రూవ్ ఇన్ ది ప్రీవియస్ థియరం, దట్ ఈస్ NPR is equal to n factorial by n minus r factorial. This formula you have to remember. Okay, NPR is equal to n factorial by n minus r factorial. Okay, this you have to remember as a formula. later. So in all these situations our R is less than or equal to n, because given number of things n is always greater than or equal to r. So the number of ways of n dissimilar things taken r at a time. So n vastuvuల నుంచి r vastuvuలు తీసుకోవాలి. That means r should be less than or equal to n. That always you have to remember. Okay. Here see that that is in particular. So suppose if r is equal to 0, it become np0. So np0 is 1. So you can substitute r is equal to 0 here. So n minus 0, n factorial by n factorial, you got 1. Okay. Similarly npn. So npn. So r substitute n here. So npn it will become n minus n. That is 0 factorial is 1. That is so that it will become n factorial. So these things you have to remember. Okay, np0 is equal to 1. Okay, npn is n factorial. So the number of ways of n dissimilar things taken n at a time. So n vastuvuలు n తీసుకొని arrange చేయాలంటే how many ways n places లో n things. So that is n factorial. Okay. Next. I have a small theorem here. See here. For 1 is less than or equal to r is less than or equal to n. Just now I say r is always less than or equal to n. Okay. npr is equal to n into n minus 1 pr minus 1. npr is equal to n into n minus 1 pr minus 1. It can easily be understood that what it says is in order to prove this, so it is very easy to prove this one. Okay, how? So say see here, npr means the number of ways of filling r blank places. Okay, using n dissimilar things. I take r blank places like this. So the first blank place can be filled how many ways? N ways. I can fill the first blank place using n dissimilar things. I can fill this first blank place with any one of the n dissimilar things. That means it can be filled in n ways. Okay. So after this, so we have n minus 1 things are left over, and we have how many of these? How many blank places are there still? R minus 1 blank places are there. So we have to fill these R minus 1 blank places using n minus 1 dissimilar things. So this can be done in n minus 1 pr minus 1 ways. n minus 1 pr minus 1 ways. Okay. So n విభిన్న వస్తువులతోటి r వస్తువులను తీసుకోగా వచ్చేటువంటి పర్ముటేషన్స్ ఎన్ని? npr. అలాగే ఇక్కడ n minus 1 ఇక్కడ one thing is used here. So one thing is over. So how many things are there? n minus 1 things are there. So using these n minus 1 things, we have to fill these r minus 1 blank places. This can be done in n minus 1 pr minus 1 ways. Okay. So according to the fundamental principle of counting, so the total work can be done in n into. So ఈ blank place ఏమో దీన్నేమో n ways. మిగతా ఎన్ని ways? n minus 1 pr minus 1 ways. The total work can be done. So n into n minus 1 pr minus 1 ways. So this is theoretical. This is the proof of this theorem using fundamental principles. And using the definition of the NPR యూజ్ చేసి ఈ విధంగా చేసుకుంటూ వచ్చాం. సో ఇలా కాకుండా మామూలుగా మనకి NPR డెఫినేషన్ యూజ్ చేసి చేయడం అంటే ఇలా చేస్తాము. వెరీ సింపుల్. ఇది చూడండి. సో ఐ టేక్ దిస్ RHS. ఓకే, ఇది అర్థమైంది అనుకుంటూ. సో ఈ విధంగా ఫస్ట్ బ్లాంక్ ప్లేస్ నింపిన తర్వాత మిగతా బ్లాంక్ ప్లేసెస్ ని సో ఎన్ని విధాలుగా మనం ఫిల్ చేయొచ్చు? n minus 1 p r minus 1 ways. This is over. మన డెఫినేషన్ యూజ్ చేసి మనం ప్రూవ్ చేయాలి అని అంటే n into n minus 1 p r minus 1 is equal to n into దీనికి సంబంధించిన డెఫినేషన్ రాసాను. npr NPR అంటే n factorial by n minus r factorial యూజ్ చేశాను. సో దిస్ ఇస్ n minus 1 factorial by n minus 1 minus r minus 1. ఇక్కడ 1 gets cancelled. Again we got here, this is n minus r factorial. So this is n into n minus 1 factorial. So yesterday I explained everything about factorial. So n into n minus 1 factorial is n factorial. Okay na? So and here is n minus r factorial. This is nothing but NPR. So this is the proof of this thing. So దీన్ని ఇలాగే కాకుండా see here అంటే ఇలా n ఒక్కటే ఇలా పక్కకు ఇలా n into అని రాసాను. ఓన్లీ n into నే కాదు, n into ఇంకా ఇలా కూడా రాయొచ్చు. ఇక్కడ రాసాను చూడండి. సిమిలర్లీ NPR is equal to n into కాకుండా n into n minus 1 into n minus 2 PR minus 2. సో ఇప్పుడు ఇక్కడ మీరు చూసినట్లయితే దీన్ని n వేస్తే ఇప్పుడు n one thing is used here. So how many ways can you fill the second blank place? So n minus 1 ways. So one thing is used. How many things are left over? N minus 1 things. We have to fill the second blank places in using n minus 1 dissimilar things. So this can be done in n minus 1 ways. So how many blank places are left over? Red 2 blank places are over. So how many places are there? R minus 2 blank places. How many things are left over? రెండు వెళ్ళిపోయినాయి కాబట్టి n minus 2 dissimilar things are there. So that's why. So this NPR can be is the same as n into n minus 1 into. So how many things are left over? n minus 2 వస్తువులతోటి ఎన్ని బ్లాంక్ ప్లేసెస్ నింపాలి? r minus 2. సో n minus 2 pr minus 2. ఈ విధంగా మీరు అర్థం చేసుకోవచ్చు. లేదంటే ఇప్పుడు దీన్ని ఇలా ఇక్కడ థియరిటికల్ గా కూడా ఇలా ముందుకు వెళ్తూ ఉంటే చూడండి. ఇక్కడ n into n minus 1 into n minus 2 pr minus 2. ఇదేంటి? ఫస్ట్ రెండు బ్లాంక్ ప్లేసెస్ లో నింపిన తర్వాత మనకి ఇలా వస్తుంది. నెక్స్ట్ నేను చెప్పిన కాన్సెప్ట్ నేను ఎక్స్టెండ్ చేసుకుంటూ పోవచ్చు. మీరు దీన్ని n వేసు, దీన్ని n minus 1 వేసు, నెక్స్ట్ థర్డ్ బ్లాంక్ ప్లేస్ ని n minus 2 వేసు. సో నెక్స్ట్ మిగతావి నింపుతాం. సో ఈ విధంగా మీరు డివైడ్ చేస్తూ వస్తే చూడండి. సో n into n minus 1 into n minus 2 into n minus 3 pr minus 3 వేసి ఇక్కడ మీరు అబ్జర్వ్ చేయాల్సింది ఏంటంటే ఇక్కడ ఈ NPR లో నుంచి ఒక n సపరేట్ చేసినప్పుడు ఈ n నుంచి r నుంచి ఒకటి లెస్ అవుతుంది. సో చూస్తున్నారా? n minus 1 p r minus 1 p r minus 1. సో ఇక్కడ రెండు n minus 1 రెండు సపరేట్ రెండు ఇంటుగా తీసుకున్నప్పుడు ఈ n నుంచి r నుంచి -2 అయిపోతుంది, రెండు డిక్రీజ్ అవుతుంది. అలాగే 3 టర్మ్స్ సపరేట్ చేసినప్పుడు n minus 1 n minus 2 ఇలాగ 3 టర్మ్స్ సపరేట్ చేసినప్పుడు సో n and r డిక్రీజ్డ్ బై 3. చూడండి n minus 3 p r minus 3. ఈ విధంగా మనకు నచ్చిన ఎంతవరకు అయినా రాసుకోవచ్చు. సో దిస్ ఇస్ వన్ ప్రాపర్టీ ఆఫ్ NPR. Don't get confused. Again later we are going to learn combinations also. Then we talk about everything. We talk about NCR. Don't get confused. The formulas of NPR and NCR. Okay. So this is. So ఇక్కడ ఒక చిన్న మీకోసం ఒక చిన్న ఎగ్జాంపుల్ చేశాను. So before going to that example, see here N5P3. సో 5p3ని ఎలా, ఫర్ ఎగ్జాంపుల్ మనకి ఇక్కడ NPR ఫార్ములా రాసాను కదా. సో 5p3 ఎలా ఫైండ్ అవుట్ చేయాలి? ఫార్ములా బేస్డ్ ఏం లేదు. 5p3 అంటే NPR అంటే ఏంటి? సో బిగినింగ్ విత్ n యూ హ్యావ్ టు రైట్ r ప్రోడక్ట్ ఆఫ్ r కాన్సిక్యూటివ్ నెంబర్స్ ఈచ్ డిక్రీజ్డ్ బై 1. సో సి హియర్ 5 తో మొదలుపెట్టి మనం ఇక్కడ r అంటే 3 కాబట్టి 3 నెంబర్స్ ప్రోడక్ట్ రాసుకోవాలి. చూడండి 5 into 4 into 3 60. Okay, this is one method. Our formula basic formula యూజ్ చేయాలి అని అంటే ఇక్కడ 6p2 తీసుకున్నాం. చూడండి 6p2 కావాలంటే ఎలా ఫైండ్ అవుట్ చేస్తాము? సేమ్ ఫార్ములా రాసాం. చూడండి n factorial by n minus r factorial. n అంటే 6, r అంటే 2, n factorial 6 factorial by n minus r 6 minus 2 factorial. ఓకే, ఇది. సో 6 ఫ్యాక్టోరియల్ అంటే ఏంటి? 6 ఇక్కడ 6 minus 2 ఫ్యాక్టోరియల్ అంటే 4 ఫ్యాక్టోరియల్ రాస్తాను. So ఇక్కడ 4 ఫ్యాక్టోరియల్ వచ్చేంత వరకు 6 ఫ్యాక్టోరియల్ కెన్ బి రిటర్న్ యాజ్ 6 into 5 into 4 ఫ్యాక్టోరియల్. ఫ్యాక్టోరియల్ మనం ఎక్కడైనా ఫ్యాక్టోరియల్ రాయొచ్చు. n into n minus 1 అలా రాసుకొని మనకి ఎక్కడ నచ్చితే అక్కడ ఫ్యాక్టోరియల్ పెట్టుకోవచ్చు. దట్ యూ హ్యావ్ టు అండర్స్టాండ్. సో 4 ఫ్యాక్టోరియల్ క్యాన్సిల్ అయిపోయింది. 6 5 3 30. ఓకే. సో ఇలా ఫార్ములా బేస్డ్ రాయండి. ఇలా లేదా ఇది ఈజీగా రాసుకోవచ్చు. మీరు చూడండి ఇక్కడ చెప్పాను కదా 6p2 కనుక్కోవాలి. ఇలా ఫార్ములా బేస్డ్గా కాకుండా 6p2 రాయాలంటే 6 నుంచి స్టార్ట్ చేసి రెండు ప్రోడక్ట్ ఆఫ్ 2 టర్మ్స్ తీసుకోవాలి. 6 2 ఇక్కడ చూడండి 5p3 5 నుంచి 5తో మొదలెట్టి ఎన్ని రాయాలి? 3 నెంబర్స్ రాయాలి. కాబట్టి 5 4 3 రాస్తాను. ఇక్కడ 6తో మొదలెట్టి 2 రాయాలి. 6 into 5 30. Okay, so this is the method how to write NPR. Okay, so next I come to a small example here. See here, if NP4 is equal to 1680 1680, find n. NP4 is equal to 1680, find n. So this is the example given in your textbook. See here what here is. So see here R is given. So we have to find n. Very simple. NP4 and argument n into n minus 1 into n minus 2 into n minus 3. Okay. So beginning with n మనకి ఎన్ని ఉంటాయి? 4 ఉంటాయి. 4 product of 4 consecutive terms. Okay. So అందుకని నేనేం చేశాను? 1680 I am trying to write this as a product of 4 consecutive integers. Because R is equal to 4, I am trying to write this as 4 consecutive integers. So కాబట్టి 168 into 10 గా రాశాను. Next this is 8 into 21 5 into 2. So 8 into 21 అంటే 7 3 is 21 5 into 2. So దీన్ని 4 consecutive integers product గా రాసాను. 8 అలాగే ఉంది. ఎందుకంటే 8 ఇలాగే ఎందుకు ఉంచాను? 8 7 consecutive కాబట్టి 8 7 అలాగే ఉన్నాయి. 3 2s are 6 into 5. So ఇప్పుడు 8 into 7 into 6 into 5 5 6 7. Okay. So this is product of 4 consecutive integers beginning with 8. Okay. NP4 is equal to NP4 is equal to 8 into 7 into 6 into 5. So what is n? N is equal to 8. Okay. N is equal to 8. దేంతో మొదలవుతుందో అది n అవుతుంది. ఎన్ని టర్మ్స్ ప్రోడక్ట్ లో ఎన్ని టర్మ్స్ ఉంటాయో అది r అవుతుంది. సో ఇక్కడ ఎన్ని ఉన్నాయి? ప్రోడక్ట్ లో 4 ఉన్నాయి కాబట్టి r is equal to 4. సో దేంతో స్టార్ట్ అయితే that will the largest integer will be the n. Okay. Similarly, here is another example for you. See here. ఇక్కడేమో r ఇచ్చారండి మనకి, ఫైండ్ n అన్నారు. సో ఇక్కడ మనకి r ఇవ్వలేదు, n ఇచ్చారు అండ్ ఫైండ్ r. చూడండి, if 12pr is equal to 1320 and 1320, find r కనుక్కుంటున్నాను. సో r అంటే ఏంటి? సో ఎన్ని ప్రోడక్ట్ లో ఎన్ని నెంబర్స్ ఉంటాయో అది r అవుతుంది మనకు తెలుసు. సో 12pr is equal to దీన్ని చూడండి ఎలా రాసాను? 12 into 110 గా రాసాను. ఓకే. సో 12pr is equal to 12 into ఈ 110 ని 11 into 10 గా రాసాను. ఇప్పుడు చూడండి 10 11 12 సరిపోయింది. సో అన్ని కాన్సిక్యూటివ్ గా ఉన్నాయి. అంటే 12pr 12 ఎందుకు రాసాను? ఇక్కడ 12 ఎందుకు రాసానంటే ఈ 12 n is equal to 12 కాబట్టి 12 మొదలవుతుంది. ఖచ్చితంగా ఇది 12 కి దీనికి 12 ఫ్యాక్టర్ అయి ఉంటుంది. సో కాబట్టి 12 తో డివైడ్ చేశాను, ఈ విధంగా వచ్చింది. ఓకేనా? సో కాబట్టి ఎన్ని టర్మ్స్ ఉన్నాయండి ఇందులో? ప్రోడక్ట్ లో మొత్తం 3 ఉన్నాయి. 12 11 10. సో r is equal to 3. Okay. So very simple problems, easy to understand. Okay. And try to observe and try to understand. So everything, each and everything, you don't miss the class. Okay. If you miss one class in between, so definitely the next concepts will be difficult for you. You cannot able to understand the things. Okay. See here students, here we have one more theorem. See here, if 1 is less than or is less than or equal to n, ఓకే, ఆర్ ఇస్ లెస్ దెన్ ఆర్ ఈక్వల్ టు అని ప్రతిసారి రాస్తున్నాను. ఎందుకు రాస్తున్నాను దట్ యూ కెన్ అండర్స్టాండ్. సో ఆర్ షుడ్ బి ఆల్వేస్ లెస్ దెన్ ఆర్ ఈక్వల్ టు ఎన్. ఓకే, దేర్ ఇస్ ఓకే given number of things n, it should be always greater. Okay, prove that npr is equal to n minus 1 pr plus r into n minus 1 pr minus 1. Okay, so దీన్ని ప్రూవ్ చేయాలి. దీన్ని రెండు రకాలుగా మనం ప్రూవ్ చేయొచ్చు. వన్ ఈజ్ యూజింగ్ అవర్ ఫండమెంటల్ ఫార్ములా యూజ్ చేసి చేయొచ్చు, లేదా మన ఎన్పిఆర్ ఫార్ములా యూజ్ చేసి నేను ఇక్కడ చేశాను. So okay, see here, that is I take RHS n minus 1 pr plus r into n minus 1 pr minus 1. Okay, this is equal to n minus 1 pr అంటే దీనికి సంబంధించిన ఫార్ములా రాశాను చూడండి. n factorial n minus 1 factorial by n minus 1 minus r factorial. Okay, I am using, see here, I am using this formula. npr is equal to n factorial by n minus r factorial. n అంటే ఇక్కడ ఏముంది, ఇక్కడ n plus n minus 1 ఉంది. n minus 1 factorial by n minus 1 minus r factorial plus r into, again apply the formula n minus 1 pr minus 1. That is n అంటే n minus 1 factorial n minus 1 minus r minus 1 factorial. Okay na? So this is equal to ఇక్కడ minus 1 plus 1 gets cancel, n minus r factorial numerator by n minus 1 factorial అలాగే ఉంది into r. Here see this n minus 1 factorial by this I write it as n minus r minus 1. ఎలా రాసిన ఉంటుంది minus 1 minus r minus r minus 1. Okay, now here ఈ టర్మ్ అలాగే ఉంది plus r into n minus 1 factorial by here n minus r factorial is n minus r into n minus r minus 1 factorial. మీకు in previously we discussed about, for example, 6 factorial is equal to 6 into 5 factorial. Okay, no? In general n factorial is equal to n into n minus 1 factorial. In the similar manner n minus r factorial is n minus r into n minus r minus 1 factorial. Okay, so now this term n minus 1 factorial by n minus r minus 1 factorial, take this as common factor and here is 1 plus r by n minus r, and now take LCM, r minus r plus r gets cancelled, we get n by n minus r. See here, so the next continuation is here n into c, n into n minus 1 factorial by n minus r into n minus r minus 1 factorial. Now n into n minus 1 factorial is nothing but n factorial, and here also n minus r minus 1 factorial into n minus r, that is n minus r factorial. This is NPR. Okay, so sometimes this type of problems may ask for 4 marks like that. So prepare to prove how to prove this one. So taking RHS and we can prove for LHS. Okay. See students, here is one example for you. Find the number of 4-letter words that can be found using the letters of the word equation. How many of these begin with E? How many end with N? How many begin with E and end with N? This is our problem. So see here. So what they are asking is, we have to find the number of 4-letter words. We have to form the 4-letter words using the letters of the word equation. Equation, see the word equation, it consists of 8 distinct letters. E, Q, U, A, T, I, O, N. Okay, these are all distinct, 8 distinct. They are 8 in number. Count all these letters. They are 8 in number and they are all distinct. So the same I write here. The word equation has 8 distinct letters first of all. So we have to form 4-letter words. And 8 lo 8 తీసుకొని అవసరం లేదు, 4-letter words మాత్రమే కావాలి. సో ఒక 4-letter word ఫామ్ చేయాలి అని అంటే మనము 8 distinct letters నుంచి 4 letters తీసుకోవాలి. First of all, we have to select the 4 letters and we have to arrange those 4 letters in 4 places like this. So this is first of all you have to understand that this is an arrangement. A word is nothing but arrangement of letters. If you change the arrangement, then you got another word. So that's why it is a purely permutation that we have to understand. The first thing, okay. So because we need the 4-letter words, so I take 4 blank places. 4-letter words and it consists of 4 letters. That's why first place, second place, third place, fourth place. ఈ విధంగా 4 blank places నేను తీసుకున్నాను. సో ఇప్పుడు ఏం చేయాలి? We have to fill the 4 letters here using these 8 distinct letters. So here see, we can fill these 4 places by using 8 letters in 8p4 ways. 8లో 4 తీసుకొని 8 letters లో 4 letters మనం తీసుకొని ఇక్కడ అరేంజ్ చేయాలి 4 places లో. So this can be done in 8p4 ways. అంటే the number of permutations of 8 distinct things, 8 distinct letters taken 4 at a time. So ఒకసారి 8 నుంచి 4 తీసుకొని అరేంజ్ చేస్తాం, మళ్ళీ ఇంకో 4 తీసుకొని అరేంజ్ చేస్తాం. This means 8p4 ways. The number of ways of filling 4 blank places using 8 distinct letters is 8p4. Okay. Thus therefore మనకి 4-letter words ఎన్ని వస్తాయి అంటే ఇప్పుడు 8p4 వస్తాయి. ఎన్ని వస్తాయంటే 4 blank places ని 8 letters తో how many ways can we fill? That is equal to, that is same as the number of 4-letter words. Okay. So thus the number of 4-letter words is 8p4. Is 8p4 means 8 into 7 into 6 into 5. Okay. That is 1680. So multiply చేస్తాం. So this is the total number of 4-letter words that can be found using the letters of the word equation E Q U A I T I O N. Okay. So next see here, then our next question is how many of these begin with E? How many of these begin with E? And ఈ 4-letter words ఏవైతే ఉన్నాయో 1680 1680 వీటిలో begin with E. We have to count the words begin with E. We have to find the number of 4-letter words that begin with E. Okay. So that's why what I am doing is take 4 places. I take 4 places and fill the first place with E. So because the word has to begin with E, అందుకని I first fill the first blank place with E. ఓకే. ఈ ఇక్కడ పెట్టేసాము. నెక్స్ట్ మనకి ఇక్కడ చూడండి ఎన్ని ఉన్నాయి, 3 blank places ఉన్నాయి. మొత్తం 8 distinct letters లో ఈ వెళ్ళిపోతే ఇంకెన్ని ఉంటాయి, 7 letters are there, 7 distinct letters are there. Using those 7 distinct letters we have to fill these 3 blank places. Okay, that I am writing here. We have to fill the remaining 3 blank places with the remaining 7 letters. Okay, 7 letters toti 3 blank places fill cheyali. How many ways this can be done? In 7p3 ways. So ఇది 7p3 ways, 7p3 ways గా ఎందుకంటే 7 ఉన్నాయి, 3 places నింపాలి. How many ways? 7 లో 3 తీసుకొని permute చేయాలి, arrange చేయాలి. That can happen in 7p3 ways. Okay. Hence the number of 4-letter words, begin with E is 7p3. 7p3 means ఇక్కడ రాసిన చూడండి, 7p3 అంటే 7 into 6 into 5, that is 210. Okay. This is the number of words begin with E. Next what we have, how many end with N? How many end with N అని చెప్పి అడుగుతున్నారు. సో దీన్ని కూడా మళ్ళీ అలాగే సిమిలర్గా నేను చేస్తున్నాను. వర్డ్స్ ఎండ్ విత్ ఎన్. ఓకే. How we have to count the number of words? We have to count the words that end with N. So for this similarly, so మనకి begin with E అన్నప్పుడు ఎలా చేసామో అలాగే end with N అన్నప్పుడు సిమిలర్ గా అలాగే చేస్తున్నాను. చూడండి, take 4 places and fill the last place with N. So because the word has to end with N, so I fill the last place with N. Okay. Now after filling this place, we have 3 places are left over, and we have 7 letters are there after using this letter N. So we have to fill the 3 blank places using 7 distinct letters. So this can be done in 7p3 ways. So 7 toti 3 places nimpali ante 7p3 ways ga nimpochu. So hence the number of 4 letter words that end with N is 7p3. Again 7 into 6 into 5. Okay. Again we get 210 words that begin with N. So this is second bit. After now the last question is words begin with E and end with N. We have to count the words that begin with E and end with N. Okay. For this, and it's not the very easy concept is whatever the condition is given that we have to fulfill the first. So you can begin with E and end with N. So I take 4 places here because 4 letter words are needed. I take 4 places and I fill the first place with E and the last place with N. Fill the first place with E and the last place with N. So after this we are left with 2 blank places. So 2 blank places are there, and మొత్తం out of 8 distinct letters E N వెళ్ళిపోతే ఇంకెన్నున్నాయి మన దగ్గర 6. So 6 letters are there, and using those 6 letters we have to fill these 2 blank places. So this can be done in 6p2 ways. So we have to fill the remaining 2 places with the remaining 6 letters. This can be done in 6p2 ways. Thus the number of 4 letter words begin with E and end with N is 6p2. 6p2 very simple. Ee 6p2 we write in chala easy. 6p2 ela rastam 6 into 5. So write begin with 6, write 2 factors, two numbers, 6 into 5. So that is 30. So this is the problem which is given in your textbook, and similar problems are there in your exercise also. You can solve all those problems on your own. Try to apply the condition whatever it is given in the problem. See here, first how many of these begin with E? So if a word has to begin with E, so first of all we have to fulfill that condition. So that's why I fill the first blank place with E. So next what is needed in second question? How many N with N? So N with N means you have to fill the last place with N. Like this you can understand this problem. N.
Welcome to online classes. I am Dr. M. Hemambara Reddy, JL in Botany, AP Residential Junior College, Nagarjuna Sagar. Today I am going to explain second year Botany, 5th unit, Molecular Biology. In this Molecular Biology lesson, Molecular Basis of Inheritance. So in this lesson, nucleic acids, that means DNA and RNA, structure, functions, I will explain. So in living organisms, DNA and RNA, these two together called as nucleic acids. DNA and RNA are the building blocks of genetic material. Genetic material means it is responsible for the transmission of characters from parents to offspring. That means from one generation to next generation. DNA is the genetic material in most of the organisms. That means generally except plant viruses, remaining organisms, DNA is the genetic material. RNA is the genetic material in some viruses. Generally, in plant viruses, RNA is the genetic material. RNA mostly functions as messengers. Generally, living organisms, we can observe three types of RNAs: mRNA, tRNA, rRNA. Later I will explain functions of RNAs. So, RNA also found in all organisms except some viruses. RNA in majority of the organisms functions as messengers. That means generally involved in protein synthesis. So DNA and RNA together called a nucleic acid. These are the building blocks of genetic material. DNA is the genetic material in most of the organisms. RNA, that means ribonucleic acid, is the genetic material in some viruses. DNA means deoxyribonucleic acid. RNA mostly functions as messengers. Structure of polynucleotide chain. Polynucleotides are the polymers of nucleotides. Poly means many. DNA and RNA are polynucleotides. That means DNA and RNA made up of nucleotides. So in DNA and RNA, so many number of nucleotides are there. That's why this DNA and RNA called as a polynucleotide chain. So nucleotide possess three types of components. That means nitrogen base. This is nitrogen base. This is nitrogen base, sugar and phosphate. These three together called as nucleotide. So this is one nucleotide, this is one nucleotide, this is one nucleotide, this is one nucleotide. So that so many number of nucleotides are there in DNA and RNA. That's why DNA and RNA called as polynucleotide chain. You see here, phosphate is a connected fifth carbon of the deoxyribose sugar. That's why this is called as a 5 prime. So 5 prime is the starting point of the DNA chain. You see here, hydroxyl group is a connected third carbon of the sugar. This is treated as a 3 prime. So in DNA and RNA, each strand starting point is called as a 5 prime, ending point is called as 3 prime. This is very important. So DNA and RNA made up of nucleotides. That's why these are called as polynucleotide chains. Polynucleotides are the polymer of nucleotides. DNA and RNA are polynucleotides. So, you see, in nucleotide, 3 components are there. Nitrogen base, pentose sugar. Sugars are different types based on number of carbons. Pentose sugar means the sugar possesses 5 carbons, 5 carbon sugar. For example, glucose, fructose, these are the hexose sugar, 6 carbon sugar. Whereas in DNA and RNA, we can observe pentose sugars. That means these sugars possess 5 carbons. So, a phosphate group. So, nitrogen base, sugar, phosphate, these 3 together called as nucleotide. So, these 3, how together, you see here, this is sugar. That means 5 carbon sugar. Okay. So, this is a nitrogen base. Nitrogen base is a connected to that is a 5 carbon sugar. And you see this is a phosphate. So, phosphate is a connected to the sugar. So, nitrogen base, this is sugar, phosphate. These 3 together called as nucleotide. So many number of nucleotides are there in DNA and RNA. That's why DNA and RNA called as a polynucleotide chain. This is structure of polynucleotide chain. Nitrogen base in nucleic acids, that means DNA and RNA. 2 types of nitrogen bases are there. Purines, pyrimidines. Purines are called double ringed nitrogen bases. You see, this is one ring, this is one ring. This is one ring, this is one ring. So, double ringed nitrogen bases are called as purines. Single ringed nitrogen bases are called as pyrimidines. So, in nucleic acids, purines are 2 types, adenine, guanine. Whereas pyrimidines are 3 types, cytosine, thymine, uracil. So, in nucleic acids, total types of nitrogen bases are 5, adenine, guanine, cytosine, thymine and uracil. DNA possesses 4 types of nitrogen bases. RNA also possesses 4 types of nitrogen bases. So in DNA, adenine, guanine, cytosine, thymine. These 4 nitrogen bases are present. Uracil is absent in DNA. In RNA, also possesses 4 types of nitrogen bases. Adenine, guanine, cytosine and uracil. Thymine is absent in RNA. That means thymine present in DNA, absent in RNA. Uracil present in RNA, absent in DNA. So, adenine, guanine, cytosine, these 3 nitrogen bases are common both in DNA and RNA. Thymine present only in DNA, uracil present only in RNA. So, Thymine, that means called as a 5-methyl uracil, present only in DNA and uracil only in RNA. So, adenine, guanine, 2 belongs to purines, that means double ringed nitrogen bases. Cytosine, thymine, uracil, these 3 together called pyrimidines, that means single ringed nitrogen bases. So total 5 types of nitrogen bases are present in nucleic acid, that means DNA and RNA. Nitrogen base is linked to pentose sugar through N-glycosidic linkase to form nucleoside. So, nucleoside means nitrogen base and sugar. The two together called as nucleoside. That means here no phosphate. So, nucleoside plus phosphate together called as nucleotide. So, nitrogen base and pentose sugar is connected with one bond, that means N-glycosidic bond. Very important for MSET and NEET point of view. So, the bond between sugar and nitrogen base, both the DNA and RNA, called as N-glycosidic bond. So, this is pentose sugar, this is nitrogen base. These two connected with N-glycosidic bond. Structure of polynucleotide chain: nucleosides in RNA, nucleosides in DNA. Nucleosides in RNA: adenosine, guanosine, cytidine, uridine. These are the 4 types of nucleosides in RNA. In DNA, deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. These are the 4 types of nucleosides in DNA. Anyhow, nucleoside means nitrogen base and sugar, that too. You see, this is sugar, this is nitrogen base, adenine. This is guanine. This is cytosine. This is uracil or thymine. That means uracil present in RNA, thymine present in DNA. So, in RNA, 4 types of nucleosides: adenosine, guanosine, cytidine, uridine. In DNA, deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. So, these are the different types of nucleosides in DNA and RNA. Structure of polynucleotide chain. So, in polynucleotide chain structure, when we observe nitrogen base plus sugar plus phosphate group, 3 together, and it forms as a nucleotide. You see, this is phosphate. This is sugar, pentose sugar. That means 5 carbons are there. So, 1, 2, 3, 4, 5 carbons are there. This is nitrogen base. These 3 together, nitrogen base, sugar, phosphate, 3 together called as nucleotide. Whereas nucleoside possesses only sugar and nitrogen base only. Structure of polynucleotide chain. In RNA, each nucleotide has an additional hydroxyl group at second position of ribose sugar. Two nucleotides are linked through 3' 5' phosphodiester bond to form dinucleotide. When more nucleotides are linked, it forms polynucleotide. So, in DNA and RNA, so how these nucleotides are connected, you see by this diagram. See, this is ribose sugar. Ribose sugar present only in RNA, whereas in DNA, deoxyribose sugar is there. So, in RNA, each nucleotide has an additional hydroxyl group at second position of ribose. Two nucleotides are linked through 3' 5' phosphodiester bond to form dinucleotide. When more nucleotides are linked, it forms polynucleotide. So, this is dinucleotide. That means this is one nucleotide, this is one nucleotide. These two nucleotides are connected with this phosphodiester bond. So, if more nucleotides are there, that is called as polynucleotide. So, this is a 5', this is 3'. 5' is starting point, 3' is ending point of each chain of DNA or RNA. When we observe the innovations of DNA, Frederick Mischer in the year 1869 identified DNA and named it as nuclein. James Watson and Francis Crick proposed double helix model of DNA. It was based on X-ray diffraction data produced by Wilkins and Franklin. That means Messrs. Wilkins and Rosalind Franklin. So, when we observe the innovations, Frederick Mischer identified and named as a nuclein. Watson and Crick proposed double helix model of DNA. That means two strands of DNA arranged anti-parallel and twisted. That structure is called as a double helix model. So, in DNA, two strands are there. The two strands arranged anti-parallel and twisted. So, that is called as a double helix model. And it was based on the X-ray diffraction data produced by Wilkins and Franklin. DNA is made up of two polynucleotide chains coiled in a right-handed fashion. Pitch of the helix, the length is 3.4 nanometers or 34 angstroms. Number of base pairs in each turn is 10. Distance between adjacent base pairs is 0.34 nanometers or 3.4 angstroms. You see, this is a DNA. This is one strand, this is one strand. Two strands are connected with hydrogen bonds. So, DNA is made up of two polynucleotide chains. This is one chain, this is one chain. It is a right-handed fashion. So, that means the right side twisted one. So, This type of DNA is called as a B-form DNA. Pitch of the helix is a 3.4 nanometers or 34 angstroms. That means one complete turn is called as pitch. One complete turn means here to here DNA shows a 360 degrees curve. Okay? Or from here to here, it is one complete turn. One complete turn means 360 degrees. Length of one complete turn is a 3.4 nanometers or 34 angstroms. That means a 10 angstroms is equal to 1 nanometer. So from here to here, 10 pairs of nucleotides are there. 10 pairs means 20. So 10 pairs of base pair. 20 base pairs means 20 nucleotides are there. Distance between adjacent base pairs. You see, this is one nitrogen base, this is one nitrogen base. The distance between two nitrogen base of the two same strands is 0.34 nanometers equal to 3.4 angstrom. Here to here, one complete turn length is 34 angstroms. The distance between two successive nucleotides is 3.4 angstroms or 0.34 nanometers. Here to here, one complete turn, the angle is 360 degrees. The angle between two successive nucleotides is 36 degrees. Here to here, one complete turn, 360 degrees. The angle between two successive nucleotides is 36 degrees. So, DNA is made up of two polynucleotide chains coiled in a right-handed fashion. Pitch of the helix is 3.4 nanometers equal to 34 angstroms. Number of base pairs in each turn is 10. Base pairs I am saying. Distance between adjacent base pairs is 0.34 nanometers or equal to 3.4 angstroms. When we observe structure of DNA, backbone of DNA is formed of sugar and phosphate. The bases project inside. The two chains have anti-parallel polarity. That is, one chain has the polarity 5' to 3' and the other has 3' to 5'. You see here, this is the DNA. This is one strand, this is one strand. That means this is one strand, this is one strand. In these two strands, in this strand, this is 5' starting point. Opposite to here, this is 3', this is ending point. So, this is one strand and this is one strand. Two strands are connected with the anti-parallel. So, this is starting point, this is ending point. Here, this is starting point, this is ending point. So, two strands arranged anti-parallel. And these two strands connected with hydrogen bonds. That means, you see, this is nitrogen base, this is nitrogen base. Adenine pairs with thymine. Guanine pairs with cytosine. That means, adenine is complementary with thymine and vice versa. Guanine is complementary with cytosine. In between adenine and thymine, two hydrogen bonds are there. in between guanine and cytosine and vice versa, 3 hydrogen bonds are there. So, 2 strands of DNA connected with hydrogen bonds, that hydrogen bonds described by the scientist Pauling. So, backbone of DNA is formed of sugar and phosphates. The bases project inside. The 2 chains have anti-parallel polarity. That is, one chain has the polarity 5' to 3', and the other has 3' to 5'. The structure of DNA, the bases in 2 strands are paired through hydrogen bonds, forming base pairs. In between adenine and thymine, 2 hydrogen bonds. In between cytosine and guanine, 3 hydrogen bonds. That means purine comes opposite to a pyrimidine. This generates uniform distance between the 2 strands. So, in DNA, adenine is a complementary with thymine and vice versa. Cytosine is a complementary with guanine and vice versa. In between adenine and thymine, 2 hydrogen bonds. In between cytosine and guanine, 3 hydrogen bonds are there. So, you see, this is adenine and thymine. In between adenine and thymine, 2 hydrogen bonds. Whereas in between cytosine and guanine, 3 hydrogen bonds are there. So, this is one strand, this is one strand, 2 strands are connected with this hydrogen bonds. So, number of hydrogen bonds in between adenine and thymine and vice versa, 2 hydrogen bonds. In between cytosine and guanine and vice versa, 3 hydrogen bonds are there. So, adenine is a purine, thymine is a pyrimidine. That means a purine pairs with pyrimidine. Cytosine is a pyrimidine, guanine is a purine. So, pyrimidine pairs with purine. See the structure of DNA. So, 2 strands. The 2 strands of DNA is not a straight one. It is a twisted one. That's why DNA structure is called as a double helical structure. It is a right side twisted structure. When we observe these 2 strands, this is one strand and this is one strand. In this strand, you see, this is phosphate group. This is phosphate group, phosphate group, phosphate group. This is sugar, phosphate sugar, phosphate sugar, phosphate. That is pentose sugar, pentose sugar, pentose sugar. That means a 5-carbon sugar. Pentose means 5-carbon sugar. These are the nitrogen bases. So, nitrogen base, pentose sugar and phosphate. These 3 together are called as a nucleotide. So, phosphate, pentose sugar, nitrogen base. So, this is one nucleotide, this is one nucleotide, this is one nucleotide. So many number of nucleotides are there. That's why this DNA is called as polynucleotide chain. So, between 2 strands, hydrogen bonds are there in between nitrogen bases. So, between adenine and thymine, 2 hydrogen bonds. Between guanine and cytosine, 3 hydrogen bonds are there. Erwin Chargaff proposed one rule in DNA. In DNA, the proportion of adenine is equal to thymine and proportion of guanine is equal to cytosine. That means always adenine complementary with thymine. Guanine is complementary with the cytosine. So number of adenines is equal to number of thymines. Number of guanines is equal to number of cytosines. So adenine plus guanine is equal to thymine plus cytosine, or adenine plus guanine by thymine and cytosine is 1. That means number of purines is equal to number of pyrimidines. That is called as Chargaff principle or Chargaff rule. So adenine is complementary with the thymine, guanine is complementary with the cytosine, so that adenine plus guanine equal to thymine plus cytosine. So this is proposed by Chargaff. That's why this rule is called as Chargaff principle. That means the number of purines are equal to number of pyrimidines. Regarding this DNA, lambda 174, it is a bacteriophage. Bacteriophage means it is a virus. Virus attacks the bacteria is called as bacteriophage. Has 5,386 nucleotides are there. That means the number of nucleotides are fixed in lambda 174 bacteriophage. Each bacteriophage possesses 5,386 nucleotides. Whereas the bacteriophage lambda has 48,502 base pairs. So this is bacteriophage lambda has 48,502 base pairs. So in this bacteriophage, DNA is a single strand. Here, DNA is a double strand, so that 48,502 base pairs are there. In Escherichia coli has 4.6 into 10 to the power of 6 base pairs. E. coli means Escherichia coli, generally found in human beings' intestine. So each E. coli possesses 4.6 into 10 to the power of 6 base pairs. That means number of nucleotides is different in different organisms. Haploid content of human DNA. That means in human beings, each cell possesses 3.3 into 10 to the power of 9 base pairs. Haploid content of human DNA. That means one set of chromosomes. So each cell in human being possesses 2 sets are there. Okay. So haploid content of human DNA is 3.3 into 10 to the power of 9 base pairs. So in human beings, all the cells are diploid. That means that 2 sets of chromosomes are there. 2 sets possess 46 chromosomes. That's why in human being, the number of chromosomes are 2n equal to 46. 2n means 2 sets. Each set possesses 23 chromosomes. In these 23 chromosomes, 3.3 into 10 to the power of 9 base pairs are there. So lambda 174, 5,386, Bacteriophage lambda has 48,502 base pairs. Escherichia coli possesses 4.6 into 10 to the power of 6 base pairs. Haploid content of human DNA is 3.3 into 10 to the power of 9 base pairs. Calculation of length of DNA. So length of DNA is number of base pairs into distance between two adjacent base pairs. Example, number of base pairs in human beings 6.6 into 10 to the power of 9. Hence, the length of DNA is 6.6 into 10 to the power of 9 into 0.34 into 10 to the power of 9. So, 2.2 meters. That means in each cell of human being, the DNA length approximately 2.2 meters. So the cell is microscopic. We can't observe a single cell with our naked eye. But in that single cell, 2.2 meter length of DNA is there. That means the DNA is highly twisted structure. That twisted DNA is located in a cell. In E. coli, length of DNA is 1.36 millimeter. That means 1.36 into 10 to the power of 3 meters. So the number of base pairs is 1.36 into 10 to the power of minus 3 by 0.34 into 10 to the power of minus 9. So equal to 4 into 10 to the power of 6 base pairs. So DNA, the length we can calculate that by number of base pairs into distance between two adjacent base pairs. So the distance between two adjacent base pairs is 0.34 nanometers or 3.4 angstroms. So packing of DNA helix. How DNA is packed in a cell. That means in human beings, already I was explained, the cell is microscopic, we can't observe with our naked eye. But each cell possesses a 2.2 meter length of DNA is there. The DNA is formed as a twisted structure in each cell. Packing of DNA helix. In prokaryotes means in some of the organisms, a conspicuous nucleus is absent. That means there is no nuclear envelope. Such a cells are called as prokaryotic cells. These prokaryotic cells are phylogenetically primitive. Whereas phylogenetically evolved cells are called as eukaryotes. Eukaryotes, the cells possess a conspicuous nucleus is there. That means nuclear envelope is there. So example for prokaryotes, bacteria and blue-green algae. These are the belongs to prokaryotes. So in prokaryotes, example Escherichia coli, the DNA is not scattered throughout the cell. DNA is negatively charged. So it is held with some positively charged proteins to form nuclei. So DNA connected with a positively charged proteins so that it forms as a nuclei. So this is bacteria cell. You see this is a nuclei. So DNA is associated with some proteins. So prokaryotic cell, this is a plasmid. Plasmid means This is a circular DNA. Whereas chromosomal DNA, it is not a circular DNA. It is a double-strand helical one. It is called as chromosomal DNA or called as nucleoid. Whereas plasmid is a circular DNA. Plasmid also double-strand structure. So in prokaryotes, the DNA is not scattered throughout the cell. DNA is negatively charged, so it is held with some positively charged proteins to form nuclei. In eukaryotes, there is a set of positively charged basic proteins called histones. Histones are rich in positively charged basic amino acid residues, lysine, arginine. 8 histones form histone octamer. Negatively charged DNA is wrapped around histone octamer to give nucleosome. You see, this is a nucleosome structure. In this nucleosome, histone core is there. This is histone core. In this histone core, 8 histones are there. So, H2A, H2B, H3, H4. 4 types of histones are there. H2A, H2B, H3, H4. So, in the histone core, 2 H2As, 2 H2Bs, 2 H3s, 2 H4s. So, total 8 histones are there. 4 types are there. Each type 2 histones are formed. This is called as histone core. Histone core is wrapped by DNA. Histone core wrapped 2 turns with the DNA. So, in eukaryotes, there is a set of positively charged basic proteins called histones. Histones are rich in positively charged basic amino acid residues, lysine and arginine. The question came several times in MSET and NEET also. So, the histones charged basic amino acid residues, lysine and arginine. 8 histones form histone octamers. Negatively charged DNA is wrapped around histone octamer to give nucleosome. So, this is nucleosome. So, this is histone core. This is a DNA wrapped 2 turns. This is 1 turn and this is 1 turn. So, here 1, 2, 3, 4, 5, 6, 7, 8. 8 histones are there. That's why it is called as histone octamer. 4 types are there. H2A, H2B, H3, H4. So, each 2 types are there. 2 H2As, 2 H2Bs, 2 H3s, 2 H4s are there. A typical nucleosome contains 200 base pairs are there. So, this is one nucleosome. This nucleosome possesses 200 base pairs. Therefore, total number of nucleosomes in human, you see, E cell possesses 6.6 into 10 to the power of 9 base pairs. So, each nucleosome possesses 200. So that 3.3 into 10 to the power of 7 nucleosomes are present in each cell of human being. So this is one nucleosome, this is one nucleosome. 2 nucleosomes are connected DNA. The DNA is called as a linker DNA or also called as basal DNA. So this is one nucleosome, this is one nucleosome. Each nucleosome wrapped with 2 terms of DNA. Between these 2 nucleosomes, DNA is there. This DNA is called as a linker DNA or also called as spacer DNA. So, a typical nucleosome contains 200 base pairs, so that in human being each cell possesses 3.3 into 10 to the power of 7 nucleosomes are there. Nucleosomes constitute the repeating unit to form chromatin. Chromatin is the thread-like stained bodies. Nucleosome in chromatin is beads on string. So this is one nucleosome, this is nucleosome, this is nucleosome, this is nucleosome, this is nucleosome. So nucleosomes constitute the repeating unit to form chromatin. So it is a twisted structure. Chromatin is the thread-like stained bodies. Nucleosome in chromatin is a beads on string. So chromatin is packed so that it forms chromatin fibers. Chromatin fibers twisted and it forms as a coiled and a condensed at a metaphase stage. And this coiled condensed structure compactly together and it forms as a chromosomes. So in a normal cell at a metaphase stage, that's why metaphase stage is the most suitable stage to observe chromosomes. So chromatin packed and it forms as a chromatin fiber, and chromatin fibers coiled and condensed and it forms as a chromosome. So higher level packaging of chromatin requires non-histone chromosomal proteins. So to form chromatin requires non-histone chromosomal proteins. So in DNA, based on staining, we can observe two different parts, euchromatin, heterochromatin. So euchromatin means loosely packed, transcriptionally active region of chromatin. It stains light. That is called as euchromatin. You see, this is euchromatin. Heterochromatin means densely packed, inactive region of chromatin. It stains dark. So generally, chromosomes are stained with acetocarmine pigment. When stained with acetocarmine, lightly stained one is called as euchromatin, thickly stained one is called as heterochromatin. Euchromatin is a loosely packed, transcriptionally active region of chromatin. It stains light. Heterochromatin, densely packed, inactive region of chromatin. It stains dark. Very important for 2 marks. What is meant by euchromatin and what is meant by heterochromatin. So euchromatin is genetically active. Heterochromatin is genetically inactive or passive. So that in chromosome we can observe euchromatin and heterochromatin regions. So up to here I was explained structure of DNA. Now the following animation you see so that you can understand structure of DNA in detail. Thank you.
Topic of this presentation is structure of DNA. First of all, we must learn basic concepts related to this topic. So we will start this presentation with nucleotides. as these are units or building blocks of nucleic acids, RNA and DNA. A nucleotide consists of three components: nitrogenous base, phosphate group and pentose sugar. As shown in the flowchart here, there are two types of pentose sugars, ribose and deoxyribose. Ribose sugar is present in RNA, whereas deoxyribose sugar is present in DNA. As shown in the diagram, a pentose sugar is a 5-carbon ring, and these carbons are named as 1' carbon, 2' carbon, likewise 5' carbon. If you know this structure is clear to you, then you will be able to understand when we say 3' position or 5' position of DNA strand in the discussion ahead. One more important point, compare the structure of ribose sugar and deoxyribose sugar. So we find hydroxyl group at 2' position in ribose sugar, whereas hydrogen group at 2' position in deoxyribose sugar. Now this presence of hydroxyl group at 2' position in ribose sugar makes it more reactive than deoxyribose sugar. Therefore, RNA is more reactive than DNA, as we already know. Now the nitrogenous bases. So two types of nitrogenous bases, pyrimidines and purines, exist. And here we have shown the structure skeleton of these nitrogenous bases. Purine is the heterocyclic ring of 9 atoms of carbon and nitrogen, whereas skeleton of pyrimidine is a heterocyclic ring of 6 carbon and nitrogen. Some modifications occur in these rings to form various types of bases as shown here. 2 types of purines, adenine and guanine, and 3 types of pyrimidines, thymine, cytosine and uracil are seen in these nucleic acids. With the help of these diagrams, we will understand difference between nucleoside and nucleotide. See this diagram of nucleoside. Here a nitrogenous base is attached at 1' position of a pentose sugar with the help of N-glycosidic bond. So when nitrogenous base is attached to pentose sugar, then this combination is called a nucleoside. And when a phosphate group joins this nucleoside, then a nucleotide is formed. Now see this structure. So here this is the pentose sugar, and at 1' position a nitrogenous base is attached through N-glycosidic bond, and at 5' position a phosphate group is attached through ester bond. So this is the structure of nucleotide, and combination of pentose sugar and nitrogenous base is the nucleoside. So this is the difference between these two units. We will now understand formation of phosphodiester bond between two nucleotides with the help of this diagram. Now see the alignment of first nucleotide. This is the 5' position of first nucleotide with phosphate group, and this is the 3' position with hydroxyl group. And the second nucleotide is also aligned in the similar way. This is the 5' position with phosphate group, and this is the 3' position with hydroxyl group. Now hydroxyl group of 3' position of nucleotide first will react with OH group of phosphate at 5' position of nucleotide second. As a result, a molecule of water will be eliminated and phosphodiester bond will be formed between 3' position of nucleotide first and 5' position of nucleotide second. So in this way we have shown you the formation of dinucleotide chain, and this is the 5' position of dinucleotide chain, and this is the 3' position of the dinucleotide chain. So we can say that this dinucleotide chain is running from 5' to 3' direction. So if our basics are clear like this, then it will help you understand and retain the structure of DNA very well. With the help of this diagram, we will now understand the structure of a polynucleotide chain in detail. So here we have shown this polynucleotide chain, and this is its 5' end with phosphate group, and this is the 3' end of this polynucleotide chain. These are the phosphodiester bonds between the nucleotides in this polynucleotide chain, and a nitrogenous base is attached to each nucleotide, and these nitrogenous bases are protruding into the right side of this polynucleotide chain. And in this polynucleotide chain, this is the 5' end and this is the 3' end. So we can say that this polynucleotide chain is running from 5' to 3' direction. So after understanding this structure of polynucleotide, it will be very easy to understand the concept of anti-parallel orientation of DNA strands in a DNA duplex. One more thing we want to tell to our students that in this polynucleotide chain, this pentose sugar and phosphate group, these two together make the backbone of this polynucleotide chain, and nitrogenous bases remain free to form hydrogen bonds with the nitrogenous bases of the other strand of DNA to make the double-stranded structure in a DNA duplex. We will now understand making of double-stranded DNA and its further coiling. Now, two strands align themselves in such a way their bases lie opposite to each other, and strands also follow anti-parallel orientation. Here, meaning of being anti-parallel is that if one strand is running from 5' to 3' direction, then the other strand runs from 3' to 5' direction. And this bases also follow complementarity rule, which means that adenine always pairs up with thymine with the help of 2 hydrogen bond, and cytosine pairs up with guanine with the help of 3 hydrogen bonds. In this way, following these rules, a duplex or double-stranded DNA is made. Now we will compare this double-stranded DNA with a rope ladder. Visualize that you are holding a small rope ladder in your hands and twisting it clockwise or anti-clockwise. Then if ladder remains straight during twisting, then it will result in symmetrical coil with equal sized grooves in its sides. But if the rope ladder gets tilted a bit during twisting, then it will result in an asymmetrical coiling with grooves of unequal size in its sides. Likewise, DNA duplex gets tilted in the same way during coiling, due to which we observe major and minor grooves in its structure. Finally, we will now understand the structure of DNA with the help of this diagram. Here, major and minor grooves are shown in the diagram, and we have already told how these grooves are formed during coiling. And see this anti-parallel orientation of strands. One strand is running from 5' to 3', while the other strand is running from 3' to 5' direction. Thickness of this duplex is 2 nanometer. And here one complete turn of DNA strand is shown, and with one major and one minor groove. Length of one complete turn is 3.4 nanometer, and there are 10 base pairs in one turn. 10 base pairs are not shown in this diagram, so please don't count the base pairs in this one. Now by calculation we find that there is a distance of 0.34 nanometer between two base pairs. So this is the structure of DNA, and we hope you have understood it very well. Please check your understanding by answering these questions.